the organizers for giving me this opportunity to come talk, so thank you very much. As Moran said, I look at the origin of life from the perspective of a physical organic chemist. And from that perspective, RNA, it's something really tough to crack, and I'm going to give you insight into that right now. So we all know about the RNA world hypothesis, this research paradigm that says the DNA protein-based biochemistry of extant life was preceded by a biochemistry predominantly mediated by RNA. But there are serious problems with this if you're talking about a prebiotic route to RNA. So I'm going to show this approach, which I've called the historical approach to RNA. Previously, it was thought that we could start with simple prebiotic precursors and build them up incrementally to form RNA. But there are many problems associated with this build-up approach. For example, we want the sugar ribose in RNA. But ribose is produced among a variety of sugars in model prebiotic reactions. So it's not immediately obvious how you would select ribose over other sugars. Furthermore, even if you could get ribose, the glycosylation chemistry required to make nucleosides does not work under prebiotically plausible conditions. So here you see cytosine. It does not form nucleosides with ribose under prebiotically plausible conditions. Furthermore, even if you could get this nucleoside, there are problems associated with phosphorylation, although we just saw beautiful work by Bradley Burkar in my lab about a method to get over that. Nevertheless, there are still problems about polymerization of those nucleotides to form RNA. So in the HUD lab, we take a different approach. This approach postulates that RNA is the product of chemical or biological evolution. And I don't think that this is too hard to accept if you're willing to accept the idea that DNA could have been the evolutionary product of RNA. So DNA and RNA are the ones that exist today. If something existed before that, it has no evidence left over. And it's a real tough challenge to figure out what the different components could have been. So conceptually, we start out by saying we're going to divide our protonucleic acid into three distinct functional units: the recognition unit, which today are the nucleobases; the trifunctional connector, which today is ribose or deoxyribose; and the ionized linker, which is phosphate today. So then we're just going to go through each one and think about what are the most prebiotically reasonable precursors that we could have had. So first we'll start with the recognition units. In a prebiotically plausible mixture, you would not just have A, U, G, and C, but a variety of heterocyclic compounds that could have supported base pairing and could have been present in your proto-RNA. So how do we select certain ones from that to get to our candidate proto-RNA? The trick is that some of them have the ability to self-sort out of this complex mixture and form supramolecular assemblies. So for example, I have highlighted here triaminopyrimidine in green and cyanuric acid in purple. These are the ones, among others, that have the ability to self-assemble. If we can get this non-covalent supramolecular assembly, perhaps we can use it as a scaffold to stitch together a polymer and create a true informational molecule. Now, there are only certain heterocycles that will support a supramolecular assembly such as these, and these four are the ones that we're most interested in: barbituric acid, TAP, triaminopyrimidine, cyanuric acid, and melamine. They form this hexameric structure, this hexad, which presents a very large stacking surface in water. And because of that, this hydrophobic surface allows them to stack on each other so that you hide most of the hydrophobic surface and you get this supramolecular assembly. In addition to that, these heterocycles have the advantage that they are very chemically reactive relative to the canonical nucleobases. So I mentioned before that the canonical nucleobases A, U, G, and C do not react with ribose to form nucleotides or nucleosides. However, TAP does, and the first demonstration of this was in our lab about 5 years ago, where when you react TAP with ribose, you get a variety of nucleoside products, the most predominant of which is the beta-C ribofuranocide. The reason that's important is because we could have ended up with many different forms of ribose, but the one that popped out in the greatest abundance was the form of ribose that is present in RNA today. Additionally, when incubated with cyanuric acid in the appropriate buffer, you can get these supramolecular assemblies, which you can actually detect by atomic force microscopy. And they have the appropriate size for this hexad structure. So it's great that it works with one nucleobase, but we need at least two to support an information system. So we turned to a different set of heterocycles. This is melamine, and this is barbituric acid. We thought these were attractive because they are structurally analogous to the extant AU base pair, which, again, cannot form under prebiotically plausible conditions. But perhaps this one can. And indeed it does. If you take barbituric acid or melamine and react them with ribose 5-phosphate in water, in both cases you get nucleotides. And if you take these crude reaction mixtures that have formed non-canonical nucleotides and combine them at the appropriate pH, you again get this supramolecular assembly appended with ribose phosphate units. And again, you can detect the presence of this supramolecular assembly by atomic force microscopy. Another really cool property of the system is that on the macroscopic scale, these assemblies efficiently prevent the bulk flow of water so that you get this hydrogel with a very high viscosity, high enough that it'll support an air bubble without allowing it to move. Okay, so that was all the work that was done on trying to elucidate what could have been the first recognition unit. But that leaves the question still of what could have been the first trifunctional connector. Because I already told you, ribose is only one component among many that are produced in the model prebiotic reaction that's typically cited for the formation of sugars, which is the Formose reaction. So it's not immediately obvious how a reactive nucleobase could have just selected ribose over any others, because from a chemist's standpoint, they basically all have the same type of reactivity via an aldehyde or a ketone. So we decided to test a set of other sugars to see what the reactivity with a non-canonical nucleobase would be. This is the set we chose. We have pentoses, tetroses, hexoses with different chemical properties. And the basic reaction that we did was to take the sugar, react it with triaminopyrimidine either at neutral or acidic conditions, and you can get a variety of products, and we would monitor all this by NMR. So here's the breakdown. Now, there's a lot of chemical structures here, but what I really want you to take away from this is that you'll see here the yields. Every single sugar worked. Every single one. That's not really that surprising from the point of view of organic chemistry, but it is kind of surprising in our field because it's often taken for granted that ribose was first. But if you can produce a variety of sugars along with ribose, and they're all equally, well, they're all reactive to an extent with TAP, then perhaps it's not a well-founded assumption that you immediately started with ribose and went straight to RNA. So we wanted to take a little bit of a closer look at what was actually forming. We did a lot of different reactions, and they're pretty messy. So we took some of the simplest ones, the ones that are based on glucose, and elucidated the structures of the products. So if you have a general glucose derivative, it can react with triaminopyrimidine to form a variety of products. These products either are linked at this nitrogen atom, or this carbon atom. Those are the most nucleophilic sites on this molecule TAP. And if you assume that the sugar is always going to be in its six-membered ring form, then there's these six possible products. These on the top have the nucleobase pointing up, that's called beta. These on the bottom have the nucleobase pointing down, that's called alpha. And these are the glucose derivatives that we tested. So when you react TAP with glucose, you get all substitutions of TAP, but you only get the beta isomers. Similarly with glucose 6-phosphate, which is a closer analog to ribose 5-phosphate. And when you try N-acetyl glucosamine, you also get these beta products, but you do not get the C glycoside. You do not get the glycoside that is connected at the carbon atom of TAP. We can talk about why that might be. My favorite hypothesis is that N-acetyl glucosamine is not as electrophilic in its protonated state as these, so it is not sufficiently electrophilic to allow for electrophilic aromatic substitution. Nevertheless, all of these have formed the beta form. So even from a chemical standpoint, there is a level of selectivity that you get that could have led to the first nucleic acids. So another thing we wanted to test with this system was, of course, the propensity for it to assemble. So I purified that compound of TAP and glucose 6-phosphate linked at the carbon atom, and I incubated it with cyanuric acid at the appropriate pH. And it was strange because it mostly precipitated, and we could see only a little bit of these tiny assemblies by atomic force microscopy. So we decided just to not even try and purify it, but start with a crude reaction mixture. So what that means is that I took TAP, I reacted it with glucose-6-phosphate. I have that reaction mixture. I'm not going to do anything to it except react or combine it then with cyanuric acid. And lo and behold, we get much more assembly. The assemblies are much longer. So first, this is really cool because this is actually the more prebiotically realistic scenario. This one required purification, intervention by a chemist. This one did not. So this system, which is more prebiotically relevant, is actually the one that supports the supramolecular assembly. It's also interesting because we don't know exactly why this one supports the assembly better, but it could be because other derivatives of TAP or underivatized TAP are also entering into this assembly and allowing for a more permissible geometry. So during this study, we had kind of an interesting result. We took ribulose, which is a ketose, not an aldose. What that means is that its carbonyl group is flanked by 2 carbon atoms rather than just 1 carbon atom and then a hydrogen. So we expected these products, right? However, that's not at all what you get. Well, they could be present, but we didn't detect them. What we did detect, however, were these what are called aldesides. They're products that could have only occurred from the isomerization of ribulose. That's really interesting because some of these are nucleosides. They're ribosides. That's important because, as I said before, ribose, it's very difficult to get exclusively to ribose in a prebiotically plausible manner. However, what if we could get to ribulose, which then has the ability to isomerize into ribose or arabinose? Turns out that there is a prebiotically plausible route to ribulose that does not produce a huge number of side products like the foremost reaction. It's this reaction of glyceraldehyde with dihydroxyfumaric acid. This is part of a so-called glyoxylose scenario, which is a hypothesis that is meant to replace or enhance the foremost reaction. So, in summary, if you take this hypothesis that RNA is the product of evolution, you can get some really fruitful results. At first, it seems really daunting because there's no evidence left of what this could have been. So it's a huge chemical space to explore. But if you impose certain constraints, you can really get some amazing results, such as that adenine and uracil, for example, were preceded by bases that were not only more reactive, but had a greater propensity for self-assembly. And this one remained, the details of this still need to be teased out, but we now know that it's not a great assumption to just go straight to ribose. We have to investigate other possible trifunctional connectors that could have been present in the first protonucleic acids. So the thing that I didn't talk about today was the ionized linker. Just to give you a little snapshot of what we're thinking, the phosphodiester is a dehydrative linkage. Perhaps it was preceded by other dehydrative linkages such as the ester or the acetal, which were more easily formed on the early Earth than the phosphoester. So with that, I'd really like to thank everybody who made this possible. Brian Cafferty was my mentor and did a lot of the work with TAP ribosides. Kim Clark was a postdoc who did all the atomic force microscopy for the TAP studies. Tyler is our new graduate student who, please go see his poster on the isomerization of ribulose and its reaction with TAP. Catherine and Megan were both excellent undergraduates who I was very privileged to mentor. Gary Schuster and Ram Krishnamurthy are our collaborators who are both excellent organic chemists. And I'd really especially like to thank Nicholas Hudd, my PI, for his tutelage and his vision. And I'd also like to thank all the members of the Center for Chemical Evolution and my lab and all of you. Thank you very much.
David, have you thought about adding some sort of short deoxypeptides or oligopeptides into your reaction mixtures, so maybe you can get some sort of catalysis and improve your reactions?
Yeah, I can see a way how that would help. We have not tried that, but I see, like for example, if you formed an ester acetal with the anomeric position of a sugar, that might help. That's a great idea. I have not tried it yet, though.
Okay, we need to move forward. Let's thank David.